Hello, my name is Aryna Taylor, and I'm here to talk to you all about my senior research project. I did my topic on determining the ability for C. elegans to make context-dependent memories. Briefly, I wanted to explain the importance of human memory as well as the importance of context-dependent memory. The human memory is a system that makes simple functions like storing and retrieving information easier for individuals. Memory helps to create an abstract representation along with updating our pre-existing memory with new information. In addition, humans are able to retain a significant amount of information and store it for a period of time, whether it be for a short period of time or for a long period of time. Context-dependent memory is when you have the ability of free recall. Some examples of context can either be their environment, temperature, and time. To give you guys a little background on the C. elegans, these are an organism that can be used as a model behavior because they resemble human beings. They possess a nervous system as well, just like humans, which is very important. They possess neurotransmitters and ion channels as well. These organisms have many advantages to them. Their small size, short life cycle, high reproductivity rate, and their ability to form new memories. The ability of free recall is present within C. elegans. These organisms are able to create memories through their context by gaining information about their environment. A study was done by Lau and many other researchers, and they wanted to determine the ability for C. elegans to apply their context-dependent memory through smell. They found out that C. elegans are capable of maintaining memory when using olfactory cues. Because of their nervous system, they are able to detect and understand their environment, chemicals, light, and temperatures. In the study that I conducted, C. elegans will be tested for context-dependent memory using temperature as a context. Within the study, there are training conditions and testing conditions. The three temperature conditions are warm, 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 cold, cold, cold. The first temperature of the condition is the training condition, while the second temperature is the testing condition. When the conditions and temperatures are the same, for instance, warm, warm, cold, cold, then the context will be the same. When the temperature is different during the training and testing, like warm, cold, then the context will be different. My hypothesis went against some of the studies that I researched that showed C. elegans being able to form memories. I hypothesized that worms have different temperatures during the training and testing will not be able to create and form new memory associations between the salt and starvation. The participants in this study were the C. elegans. They were all fed and housed at room temperature. During this experiment as well, they were young adults. They were not young when they were tested during this experiment. The materials needed for this experiment were petri dishes that contained NGM and E. coli for food, 100 millimeters of NaCl, along with 10 millimeters of NaCl. Testing plates that contained the chemotaxis agar in 2 out of the 4 quadrants, and lastly, an incubator set for 15 degrees. For the procedure, the C. elegans were trained in warm, warm at room temperature and will be put in the tubes containing NaCl for 15 minutes. Once they're placed on the petri dish, 2 quadrants that contain salt, while the other 2 quadrants did not contain salt, these C. elegans were placed in the middle, and after every 10 minutes, we would count to see how many of the C. elegans would go in each quadrant. The same procedure was replicated for the other 2 temperature condition groups as well. After all groups were tested, a chemotaxis index was taken. A chemotaxis index is seeing what % of the worms like the salt. For my results, I created a line graph to be able to show the differences between the conditions. For the warm-warm condition, there is a steady increase. However, for the cold-cold condition, it is stagnant. In my overall findings, I found to be that there was no significant difference between time and the chemotaxis index. The p-value was 0.155, showing that it is low, but not low enough. After looking at my findings, I determined that my hypothesis was not supported. In other studies, context was able to play a role in C. elegans' ability to form memories. However, it was present in my study. My study differed from the study done by Dallett and Wilcox, showing that with the change in environment, the C. elegans were able to adapt and create new memories. However, my results do mirror the study of Godin and Badley, showing that with the change of environment, the ability of free recall was impaired by the change due to the environment. For limitations in the study, there was not many, but just a few. The first is addressing the issue of time. The worms did not seem to react with the time intervals that were given. The second limitation is cleaning the petri dishes off when there was an excess amount of fluid on the dish. I would soak up the remaining fluid with a Kintec wipe, but there was a fear that while I was doing that, I was collecting C. elegans on my wipe as well. Lastly, the amount of trials proved to be an issue due to the low p-value. It was low, but not low enough to cause a significant difference. With improving this study, we want to address the limitations that were stated previously. By addressing the limitations, this could potentially create significance when being tested next. Instead of manipulating temperature, there are other variables that could be manipulated: time and the growth of the C. elegans. Thank you for all your help with this project.